Now again, I will share. Yeah, so I was talking about that membrane potential is the electrical charges or electrical force which is present across the cell membrane or at the cell membrane when the cell is at rest, not performing any activity. During rest means it is not performing any activity. So that is called as the resting membrane potential. But why this force or why this electrical charges are created at the membrane? We have to know the causes. We have to know why it is happening. Okay. Let me show you the another slide. Where is the slide? Okay. So in a normal cell, we say, let's say this is the cell membrane. This is inside of the cell and this is outside of the cell. Inside of the cell, there are some ions like potassium, sodium, chloride, phosphate, PO3 or PO4, PO4, and also protein, Similarly, outside also there are sodium, potassium, chloride, and many more are there. Calcium are there. Because of the unequal distribution, inside maybe some are more, outside there are some others are more. Because of this unequal distribution, this is equal, we said unequal means like this. So because of this unequal distribution or separation of charges, whenever you put electrode inside the cell and connect with a galvanometer and with the other electrode outside the cell, it will show you some current. It will show you some current or electrical potential. We call it as current or potential. This is called as resting membrane potential, RMP. Here it is written due to difference in concentration and permeability, because of some are more permeable going in and out, some are less permeable, and because of the concentration, many are more here and less here. Because of unequal distribution and separation, there appears to be some electrical activity which is measured in terms of millivolt, current in terms of millivolt, not like this, it will be like this, millivolt. Millivolt current will be produced at the cell membrane, where you will find more negativity inside and more positivity outside, because they are lining in such a way that the positive ions will be lined outside the cell and negative ions will be lying inside the cell. You are getting my point now?
Negative more inside, right?
Yes. Suppose this is a cell, now square shape. So more or all the negativity is lying inside the cell and all the positivity lying outside the cell. And if you connect with a galvanometer, it will show you some current, some millivolt. And this is called as resting membrane potential. If you put one electrode here, sorry, you put one electrode here and another electrode. Oh, this is because of maybe as it is showing the window, that's why. So let us connect it here. One electrode out and one electrode in, and you connect with a galvanometer. It will show you some deflection, some current will be shown as if you are checking in the switch or socket in your house. Okay, if you check with the galvanometer, it will show you some current. So similarly, in our body, all the cells will have some sort of current at the membrane, here at the membrane, there is some sort of current is there, which is called as, we call them as potential. We call them as potential or QA. Potential or QA. So this potential or QA or kaharba, this is because of why it is, because some are more permeable and some are less permeable, and it creates some concentration difference. Concentration difference. More are potassium and less potassium outside. More sodium outside and less sodium inside. More protein inside and less protein outside, like that. Because of unequal distribution, and because of that, and because of the permeability, there appears to be some sort of current is developed, and this is called as resting membrane potential. Resting membrane potential. How much is the quantity? What is the quantity of resting membrane potential? Is it 1 millivolt, 2 millivolt, 3 millivolt, something like that? It is about minus 90 millivolt in most of the cell, and
Except
minus
for
70 millivolt in neuron. Please remember, it is always minus 90 millivolt or plus or minus something, but it is near to minus 90 millivolt in almost all the cells except neurons. Neurons has got minus 70 millivolt, plus or minus something. Okay. Are you getting my point?
Yes,
Because of unequal distribution of the charges, because of the permeability difference, because of these two changes, there appears to be some potential, and this potential is known as resting membrane potential because it is at rest, because the cell is not active. This cell is normal or in a state of non-action. If the cell goes in action, we will have some different activity, which we will discuss it later on. So when the cell is not performing any active function, regular function, and it is performing regular inside and outside or permeability function or gas exchange or maybe production of food or excretion of the waste product. All these functions are vegetative, baseline function. They are not active function. Whenever cell goes in action means muscles getting contraction or the cells, the neurons getting sending the signal. These are called as activity. But when the cell is not sending any signal, not contracting, we call them as at rest. So at rest, the force or electrical charge which is present or found on the cell membrane is known as resting membrane potential. This is the simplest definition, simplest explanation of resting membrane potential. And the value is almost all the cells will have minus 90 millivolt except neurons, which has got minus 70 millivolt resting membrane potential. Remember, don't forget anything. It ranges between minus 90 to minus 70. Here, minus 70 is only in neuron, whereas minus 90 is in other type of cells. Maybe RBC, maybe skin cell, maybe epithelial cells. All these cells will show you minus 90 or muscle cell, but the neurons will show minus 70 millivolt at rest. This is called as resting membrane potential. So here is the diagram showing you that here is the membrane, and where is the inside and where is outside? The yellow color is inside. This is inside of the cell, and this is outside of the cell. There are 3 positive ions, there are 2 negative ions. Here also 3 negative ions and 2 positive ions, something which is having different. So you will see here 10 positive and 10 negative. Inside is also 10 positive and 10 negative. So at this point, the potential will be 0. There is no potential because both of them are equal. Both of them are equal. Our first condition to have force is unequal distribution and different permeability is the cause of membrane potential. So here Both of them are equal. That's why no potential. There is no force or no potential. Second example, B. There are 15 positive ions and 10 negative ions outside, whereas inside it is 5 positive and 10 negative ions. So there is some potential because of unequal distribution. Here total number is 25. Here total number is 15. So 15 different in the number of ions. Also difference in the positivity and negativity. That's why it will have potential. Because of the potential difference and because of the arrangement, the positive ions will lie outside the cell. As I told you, if it is a cell, positive ions will be aligning themselves in at the outside, whereas negative ions will align themselves inside the cell. Okay. E1 potential will be of different magnitude. Different, you can say magnitude means what is magnitude? Quantity. You can say quantity of electrical difference can be small or big or medium. So here is the example in the third, that is in this picture C. Here plus 3 ions and minus 3 ions. So there will be some potential. Here plus maybe 1, 2, 3, 4, 5, 6 plus ions and 6 minus ions. 6 and 6. Here 3 and 3. 3 negative, 3 positive. And here you will have more. Maybe 8, I think. 8 plus and 8 minus. So if you compare these three pictures, the potential is less here, potential is maximum here, potential is medium here, based on the number of positive and negative ions. Now this diagram shows you which side, how many, what is the quantity of each ion. So inside of the cell, if you remember, at least remember 2, 3 molecules like sodium, potassium, phosphate, and protein. At least remember about these elements, where they are more and where they are less. Inside is how much their concentration, and outside is how much their concentration. At least sodium, potassium, phosphate, and protein. At least remember the number of these. What is the concentration of sodium inside the cell? This is inside. Inside the cell is 14 millimole, whereas outside is 142 millimoles for sodium. This is for sodium. Remember, sodium is more outside and less inside. How much? It is 14 millimole inside and 142 millimole outside. Remember at least these numbers: 1, 2, 3, and 4. At least this you have to remember in terms of millimole. Coming to the potassium. Potassium is 140 millimole inside the cell and 4 millimole outside the cell. Whereas phosphate, 75 millimole inside the cell and 4 millimoles is outside the cell. Proteins are 40 millimoles inside the cell and 5 millimole outside the cell. So at least remember, because of this unequal distribution, you will see unequal. 14 here, 142 here, 140 here, and 4 here. So they are not equal. They are unequal distribution. That's why, because of unequal distribution and their difference in the permeability, these there will be some potential or some energy will be there at the cell membrane. We call it as resting membrane potential. The value is minus 90 millivolt for all the cells except neuron. Neuron has got minus 70 millivolt. That's it. This is the definition and information about the resting membrane potential. Now, why it happens? Why unequal distribution happens? Because of most important one is sodium, potassium, ATPase pump. ATP is pumped. Because of this pump, unequal distribution of the ions occurs. What this sodium potassium ATPase pump is doing? Sodium potassium pump is active throughout your life in all the cells, and it is sending 3 sodium ions outside. It is throwing 3 sodium ions, 1 sodium, 2 sodium, and 3 sodium outside, and it is taking 2 potassium inside. In one cycle, it is doing like this. So you can see here, 3 positive ions are going out, whereas 2 positive ions are coming in. So how much is the loss? Loss of 1 positive ions. Loss of 1 positive ion from inside to the outside. That's why inside is becoming negative. And also proteins are there. Agreed? So resting membrane potential is established. Established means, what is meaning of established? In Arabic, what we call it? If something is established, you are establishing a building.
تأسس يا دكتور. تأسس أو يتأسس.
So, yeah, something you are making here. Maybe you are making a building here. Let's say this is building is established. Al-Marifa building is established here. So this is called as establishment, or it is done here. And it is done, this resting membrane potential is established because of sodium potassium ATPase pump. Okay. So cause of resting membrane potential is establishment is sodium potassium ATPase pump. So inside becomes more potassium, outside becomes more sodium. And because of the passive diffusion, they will try to come in and the potassium will try to go out. This is the same picture showing you how it is happening. So in a cell, this sodium potassium
Doctor?
ATPase pump. Yes.
Are we going to have pictures in the exam?
Yes, in the exam, you may be having a picture showing you this picture without the numbers, maybe without number, or maybe X. So how many ions are going out of the cell of sodium? Maybe something like that. Okay.
So the doctor, the only pictures might come from resting membrane potential and active potential, huh? From these two sides only.
In action potential also there are graph. There is a graph, sorry.
So
Like
one graph from, okay.
Yeah.
Okay.
Whatever may be the question or, or type of question, first you have to read the question. It may be a picture may be supporting one. But if you read the question, you will understand what the question is asking about. Does it require a picture or not? The picture may be supporting. You cannot say that it is only the picture which will be responsible for the answer. No, it's not like that. If you know the procedure, why sodium is more inside and more outside and less inside, if you know, then you will easily understand that there is a sodium potassium ATPase form. So this picture is showing the same thing, that in every cycle, this cycle is going on. In one turn, it is throwing 3 sodium out and taking 2 potassium in. This is the function of sodium potassium ATPase pump. Why it is called as ATPase pump? Because ATPs are broken down here. Energy is utilized. ATP is broken down into ADP and inorganic or phosphate. This phosphate is utilized for this moment. This is the energy. The energy is coming from ATPs. That's why the name of the pump is sodium potassium ATPase pump. This is just like an enzyme. There is an enzyme which is breaking down ATP into ADP, and the energy is produced, and this energy is utilized to throw 3 sodium out and take 2 potassium in. And this is a type of, when energy is utilized, which type of transportation it is? When energy is utilized, it is a type of active transport. Active transport from the previous lecture. So it is a type of active transport. Okay, so here we are saying that more sodium, more potassium is there inside, and less, and more sodium is outside. And because of that, they have got a tendency to move towards the from higher to lower side. Whenever there is a difference, it is trying to move from higher to lower side. If there is a difference. So they will try to move like this and create a positivity outside and negativity inside. So maybe instead of taking the both sodium and potassium, let us take the example of potassium only. Suppose the cell is having only potassium ions, which are more inside the cell and less outside the cell. So it will try to move to the outside. When it is becoming more outside, because positive ions and positive ions, if positive ion going toward the positive ions, what will happen? They will attract or repel?
Repel.
Yes, they will be repelling. So it will try to repel. Now it will not go. More, all are positive here. And again, positive ions will go. So they will stop. They will stop the movement. Okay. So next step is there will be a point will come when no more potassium can be going out of the cell, because they will be having more positive here and also positive ions are going. So there will be stoppage. If there is a stoppage, that time the concentration of potassium will be equal to from inside to outside will be equal. Same number. Suppose 40 potassium are inside, 40 will be outside. So they will become equal. This potential, we call it as equilibrium potential. A point will come when the number of potassium ions inside and outside will become equal, and that time it is not possible for any further potassium to go out because of positivity. They are repelling each other. This is called as equilibrium potential. We call it as EK for potassium, equilibrium potential for potassium ion. Okay. This is
Doctor, when this case happened?
Hmm?
When this case happened?
We are assuming this is not happening actually. We are assuming. This is our theory, assumption, that if there is only one potassium ions are there, then it will move from more side to the lower side, higher side to the lower side. A point will come when both sides will become equal. At this point, the equilibrium potential will be achieved, and there will be no net movement. No net movement. Okay.
So can I say, like, if it's equilibrium at potassium, it's more at rest?
No, first we have to think that the cell is having only potassium ions, and we know that potassium is more inside. So suppose if it starts to go out, it will go on till the potassium ion concentration inside and outside will become equal. This is at this point, if you measure the electrical charges, it will show you a force or current which is known as equilibrium potential. Okay.
Okay.
Similarly, if we think that no, only sodium are there in the cell or outside the cell. So you know that sodium is more outside the cell, 142 milliequivalent or millimole. So they will try to go from higher to lower side. So what will happen? They will start to move from higher to lower side. Now positive ions are moving towards the positive ions. A point will come when positive ions and positive ions, they will be repelling each other. Positive here and positive here. So they will not attract. rather they will repel and they will stop the movement of sodium ions. They will stop the movement of sodium ions. At this moment, we call it as potential will be called as equilibrium potential for sodium.
But Doctor, I have a question.
Yes.
We said in the sodium, it is in more inside. So in sodium, it's
Outside.
more outside, huh?
Yeah. Yes. It is more outside. So as I told you that whenever there is a difference, it is going from higher to lower side. Okay. So it is higher here. So it will try to go inside. A point will come when both sides will become equal. Sahih?
صحيح.
So at that point, if you measure, the equilibrium potential is known as equilibrium potential for sodium. So similarly, whatever the ions are there inside and outside the cell, we have to measure the equilibrium potential for them, all of them, for potassium, for chloride,
Doctor?
for phosphate, for protein, all of them we have to measure and then combine together to know exactly how much is the resting membrane potential, right? This is the actual way of finding out the resting membrane potential. But we will not do it. I am just giving you example of sodium and potassium. Let us see now what this slide says. This slide says that at rest, when the cell is at rest, not performing any active function, at that time it is 20 to 30 times more permeable to potassium. At rest, when the cell is not actively performing any action, it is 25 to 30 times more permeable to potassium rather than sodium. It is not sodium, it is the potassium which is more permeable. And that's why the resting membrane potential is mainly due to potassium. Mainly due to potassium. Here is the equilibrium potential for potassium. Here is the equilibrium potential for sodium, which is plus 60. Remember, plus 61. Here it is minus 94. How much I told you for resting membrane potential? I told you,
negative 70, negative 90, Doctor.
yeah, minus 90 for all the cells and minus 70 for the neuron. So it is near to what? It is near to which number? Sodium number or potassium number?
Potassium.
Yeah, so it is the resting membrane potential is closest to the equilibrium potential for the ion which is having highest permeability. 25 to 30 times for potassium. Permeability for potassium is 25 to 30 times more than the sodium. That's why it is more towards minus 94. Exactly, it is minus 90 for all the cells and minus 70 for the neurons. Getting my point now?
Doctor?
Yes.
Only this much information you must know how the resting membrane potential is established and why it is minus 90, why not plus 60. Okay?
Doctor?
Yes.
Can you tell us at the last the most important site?
Most important slides?
Yeah.
It cannot be because this is one slide is telling one step, second slide is telling second step, third slide is telling third step.
Tell us the most
You have to
important.
make the picture in your mind. There is no slide. Question will not be from slide. Question will be from concept.
Okay, tell us the most important slide.
My dear, the slide will not be one slide. If you read this slide says resting membrane potential Vm minus 90, Vm minus 70. What is this? It
But
will
not
tell you
everything
nothing
can come.
unless what I explained you in last 15 minutes. If you remember and if you understood that, it can be 3, 4, 5 questions I can ask you on that. Not on one slide.
But not every step is gonna come, right?
How can it be possible? Is it the only physiology lecture this is coming in exam? There are many lectures.
Yeah, I
We
know.
usually
But
give you.
too much. If you
No, no,
can,
no.
like,
Like
if you,
a university student in university, and you are learning MBBS course, there is no like story which gives you one paragraph and there is a question and answer. No, it is the whole book or whole chapter. The concept will be checked by asking question.
I
How
know,
is it
but we're
possible?
in level 3 and like it's too much.
It's
If you
not too
can,
much. If you
if you can have
understand up till now 15 minutes, if you did not understand anything, you cannot answer anything. If you have understood the procedure, you can answer 4 to 5 questions on this.
okay.
Have you understand up till now what I told you in from
Yes,
how what is
yes,
the meaning
Doctor.
of resting membrane? Yeah.
Yes, Doctor. Continue, please, Doctor.
Yeah. What is mean by resting membrane potential? Who contribute in the resting membrane potential? Why it is minus 90? Why it is established? Why more close to this potassium, not to the sodium? These are the questions which I can ask you. So it is saying why it is close to the equilibrium potential of potassium, because potassium is more permeable than sodium. Now, resting membrane potential in unstimulated cell is called as the resting membrane potential. When the cell is not stimulated, we call it as resting. At the stage, the condition is known as resting, not active. Okay. So normal value exists between minus 70, which is only for neuron. I'm telling you many times now, and minus 90 for other cells, like maybe muscle or maybe any other tissues or any other cell. So why resting membrane potential is produced? Because of unequal distribution of ion, unequal permeability, and one important pump is sodium potassium ATPase pump. Resting membrane potential is established because of three things: unequal concentration, unequal permeability, but the most important one is the sodium potassium ATPase pump, which is what it is doing. It is throwing 3 sodium out and taking 2 potassium in. Because of this,
Doctor,
there is unequal distribution. Yeah.
Doctor, I have a question. I remember in this lecture you said the ATP's pump, it has the least role, but when generated, it plays the most important role. Is it important in the exam to know this information? Yes or no?
Yes. Three things are there for resting membrane potential: unequal distribution of ions, unequal permeability, and sodium potassium ATPase pump. These are the 3 main causes of resting membrane potential. Out of that, the most important one in the form of a pump is sodium potassium ATPase. Got it?
What's the
Yes.
last thing you tell us?
Hmm?
Like the sodium potassium pump, it's the most important.
For establishing resting membrane potential.
Okay, thank you.
Although it is making a small direct contribution, but it is most important. Okay, nerve and muscle cells are called as excitable cells because they can be stimulated. If you put a current or if you stretch them, they will respond in the form of in the form of contraction or in the form of sending the signal. So 2 types of cells are excitable cells. One is nerve and the second one is the muscle. So nerve and muscle cells are excitable because they have the ability to change their resting membrane potential, minus 90 or minus 70, to suddenly change to different levels. It may change to maybe plus 10 or plus 30. It may change to plus 10 or plus 30. So they have got the ability to quickly reverse the potential, resting membrane potential. That's why they are called as excitable cells. Nerve and muscles can change their resting membrane potential very fast to a large degree. That's why they are called as excitable cells. This is the same thing which I told you. Nerve and muscles can rapidly change their permeability. They can change their permeability rapidly, and they can change their potential. Big change in their membrane potential. That's why they are called as excitable cells. Remember, resting membrane potential of the neuron is minus 70 millivolts. Okay. All the cells membrane, all the living cells, not only only muscle and nerve. All the cells will have membrane potential where the cell is negative inside and positive outside. Nerve and muscle cells are called as excitable tissue. This is a summary. And they can change their potential to form the impulses. Nerves can change their potential to form or to generate the impulse. Or the muscles, they can change the potential to cause contraction. For muscles is the contraction. Their activity will be contraction. Okay. Some terminologies you must know.
Doctor, can I ask a question, please?
Yeah, please go ahead.
Yeah, so muscle cell cause contraction because it is excitable, so that's why they cause the contraction.
No,
Is that
can
why
you
they
repeat again?
muscle cell cause contraction? Is the
Yes.
contraction caused because they are excitable?
Yes.
So nerve cell doesn't not, but except muscle cell, they cause
Nerves,
contraction.
yeah, nerves, they don't contract, but they can generate impulse in the
Generate
form of action
impulse.
potential. That's why they are called as
Okay.
excited. Yeah. So up till now, you have seen that the membrane potential is about, let's say, minus 90. Let's say for other cells, not the neuron, minus 90. If this minus 90 is there, we call it as resting membrane potential. If this becomes less negative, yani it becomes, let's say, it becomes minus 50. Let's say it becomes minus 50, less negative. We call it as depolarization. When negativity decreases, okay, it's called as depolarization. D for decrease in polarity, D for depolarization. If it goes back to the minus 90, we call it as repolarization. Here it is written. Membrane returns to the resting membrane potential. Suppose if I draw a graph here, here it is minus 90, here it is 0, here it is plus 10 or plus 30 maybe. Okay. So if the membrane is going towards the 0, it is called as depolarization. If it is coming back towards the resting, RMP, it is called as repolarization. And if it is becoming more negative, more than the resting state, minus 90, it may be minus 100, suppose here. This is called as hyperpolarization. This is hyperpolarization. Any difficulty in this?
I didn't understand the polarization, Doctor, the first one.
The polarization is the state when the membrane potential other than 0. It's not 0. Our resting membrane potential is minus 90. So that's why it is called as polarized. Okay. Resting membrane potential or polarization is same thing. Polarized. Okay.
Doc
If it
said
is,
polarization must be negative or it can be positive.
which
Yani
one?
it can be 20.
Yes, yes,
positive.
it can be. Yes, sure,
Okay.
sure. Polarization is anything other than 0. So these are the terminologies used for showing you what is the change in the current or potential. Same thing is shown here in the form of diagram, where minus 70 here, minus 70 means which cell it will be?
The repolarization, Doctor.
No. Why it is starting from minus 70? But can you guess
As
which
it-
cell it will be?
It's شو اسمه neuron cell resting membrane.
It will be a neuron. Previously, I shown you minus 90. So it may be a muscle. It may be a muscle because I shown you minus 90. But if it is here, minus 70, you can guess. Minus 70 means it is a neuron resting membrane potential. So if it is becoming less negative, going up, this is called as depolarization. If it is coming back to the original resting state, this is called as repolarization. If it is going below the normal resting membrane potential, more negative, it is called as hyperpolarization. So excitability is after depolarization. When the cell is getting excited, it depolarizes and come back to the normal state of repolarization. So same thing is shown here in this diagram also, that excitability means when the cell is going in the direction of less negativity, losing the negativity. That's why it is called as excitable cell. So muscle cell and neurons, they are called as the excitable cell. Anything above zero is also known as overshoot. Anything above zero is called as overshoot. So suppose I draw a diagram here from minus 90 like this and coming back, and here it is the zero. So this part is the overshoot. This part is depolarization. And this part is repolarization. And this part is hyperpolarization. Okay.
Doctor, I have a question. Is threshold important? Yes or no, Doctor?
Threshold will come in the next lecture and the next action potential. It will be important, but it is in the mostly it is related to action potential. I will discuss that also.
Okay.
So as I told you, from minus 70 or minus 90, it can go like this and come back, or it can go like this and come back. So this is small change, right? This is small change. This is called as graded potential. Short distance, it will not travel from one area to other area. It will be only local potential. Graded potential is change in the potential, little amount for very short distance locally. Whereas action potential is great change in the membrane potential and it will travel. So suppose this is a neuron and I will draw it here. These are the myelin sheath. Suppose this is a neuron. So if the potential change occurs, it will be from each node to node. There, sorry, here, 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 and then go to the next neuron also. It will go to the next neuron also. This is called as, it is traveling to a long distance. This is called as action potential. But if it is maybe in the receptor, if it is only changing little bit and not traveling, this is called as graded potential. Graded means from minus 70 to minus 50, 68 maybe, or maybe minus 65, like this, very small. But action potential means from minus 70 to plus 10. or plus 30. Large change in the potential, and it is traveling from one area to other area. This is called as action potential. So again, action potential is produced, or local potential is produced because of the permeability. If the cell more permeable to some ions and less permeable to other ions. Also the type of channel present, like some channels will open. This is voltage dependent, is because of change in the voltage they will open or close, or maybe they will open or close because of some receptors. Let's say this is a channel and another channel, maybe another door. This will open if it is changed from -70 to -68, suppose. So this will open. This is called as voltage gated because of change in the voltage. This channel will open because of attachment of something here. Let's say acetylcholine is attached. So this will open and will allow the ions to move. This is called as ligand gated channel. So two types of channels are there, which is causing the potential to change either because of change in voltage or because of attachment of some chemical, some ligand. Are you
Yeah,
getting my
Doctor.
point?
Yeah, yes. So I only need to know the channels, the ionic base, membrane permeability, voltage dependent channel, or ligand gated. But ion
Yes.
channels, yes, okay.
So ion channels will open or close because of two things, either change in the voltage or because of attachment of some chemical. That's it. So this was the resting membrane potential. Now we will move to the next lecture, that is action potential.
Doctor, I want to ask you.
Hmm.
Did you record this lecture?
Yes, I am recording. Let me share another one. Action potential. Can you see the new slide now?
Yes, Doctor.
The second lecture which is coming in the exam is the graded potential or action potential. We have discussed few things in the previous lecture. Similar things will be there now. So as you know, there are two systems which control the body. One is nervous system, another one is endocrine system. Nervous system is controlling with the help of nerve signals. They are sending action potential impulses from the brain to the organ, or from organ to the brain. And this is how it is controlling. Whereas endocrine system is controlling by releasing the hormone. So nerve and muscle cells are called as excitable cells. Why they are called as excitable? Because they can change the membrane potential very quickly and to a large degree. From -90 in the muscle, they can change it to +10, or from -70 millivolt in the neuron, they can change it to +10 millivolt very quickly. That's why they are called as a excitable cell. We have seen two types of electrical change can happen. Either it will be a small change. This is called as graded potential, or it can be a big change in the membrane potential. We call it as a action potential. Action potential usually goes from -70 or -90 to plus. They will be crossing zero line and they will be reaching to +30 millivolt in this much extent. And it is not only at one place. Once it is produced, it will be going from one area to another area. In the muscle also, or in the nerve also. That's why it is traveling from one area to another area. This is the property of action potential. Action potential is traveling, whereas graded potential is just coming and going. That's it. It will not go from one area to other area. So graded potential means from -70 to -60, a 10 millivolt change. It can be due to sodium entry into the cell, and these graded potential can be combined. Suppose here is 10 millivolt change, here is 10 millivolt change. If they are near to each other, it can be look like a bigger graph. That means number 1 is combined with number 2. So that's why it will become slightly bigger. This is called as summation or addition. So graded potential can be combined. 1 plus 1 becomes 3. 1 plus 2 becomes 3. Okay, like this. So why it is happening? It is happening due to the opening of sodium channels. So sodium channels will open, or any positive ion channels will open. This will allow
Doctor?
sodium to move. Yes,
The screen is freezed.
freeze.
Like we're still in the first slide.
Okay, again I will reshare. Thank you for reminding. Oh, this is membrane potential. No, not this one. Can you see now?
Yes.
Let me make it full screen. Can you see the graded potential slide now?
Yes, doctor.
Okay. So I was telling you graded potential is a from grade 1 to grade 2 or grade 2 to grade 3, like that. It is just a small change. So if you add them together, it will be a addition will be possible here. A small change here and nearby small change, both will combine to give you the bigger graph. This is called as summation. This property is known as summation or addition. So suppose here it is 2 millivolt change, here is 3 millivolt change. This will give you a 5 millivolt change, something like that. And it is a locally present. It is present only in this area only, not traveling to other area. So here is the example showing you the graded potential will occur because of the extracellular is more positive, intracellular is more negative. Some of the positive ions move inside to cause depolarization, and this depolarization will spread to the local area only. It will not travel throughout the length. Because why it will not travel? Because after some time, this channel will be closed. This channel will be closed, and that's why there will be no more positive ions moving in. That's why it is called as a local potential or change in the potential with a small magnitude. This is why it's called as a graded potential. When you have a stronger stimulus, it will remain open for long time. If you have a, let's say this is a sodium channel. If you open it by force of the smaller force, it will close after some time. It will become closed after some time. But if you apply a greater force, it will remain open for long time, and it will take long time for closure. So that's why the potential will be bigger here, potential will be less here. So you have to remember that graded potential occurs in small size specialized region only, in a local area only of the excitable membrane, and it will be varying depending upon the force by which it is happening. If the force is small, the opening of channel will be small, and the potential will be changed in a very little quantity. So here in the center, if you draw a graph, you will see that more change will be found in the center and less change will be found at the periphery. That's why it will not spread to a large area. Now you can see, as it is written here, important. This slide is important. That means it is summarizing everything. Graded potential is localized, present only in one area, not traveling from one to other area. It can be summated, means it can be added together. You can add them together. So if the stimulus is longer, there will be a graded potential for long time. Whereas it will be dying down or decreasing in the after very short distance. It is not traveling a long distance. It is here it will be less. Here in the center, it will be more. Now, example of graded potential are the potential which is recorded in the receptor. What does it mean by receptor? Like touch receptor, pain receptor. This is the receptor. So it is recorded here in the receptors, or it may be recorded in the pacemaker cells of the heart. It is also recorded in the pacemaker cells of the heart, or it may be recorded in the end plate of the muscle end plate. Graded potential can be recorded in the muscle end plate. Muscle end plate, yesterday we discussed. This is the area where the neurons is touching to the muscle. So this area is known as motor end plate. So this area will show you graded potential. So those who are asking me which slide is important, this slide is important because it is summarizing everything what we have discussed in the previous slides. Now coming to the second part of the lecture, that is action potential. Up till now, we have seen only a small change in the membrane potential, which is called as graded potential. What is mean by action potential? It is a brief, very short, but rapid, very fast. Short and fast and large, almost 10-100 millivolt change in the membrane potential is called as action potential. So if you want to draw the graph, it will be just like a spike, because this is the time. Here on the x-axis it is the time, here is millivolt, on the y-axis it is millivolt. So time is very short from here to here. This is the time of action potential, which may be 0.1 second, maybe 0.1 second. So very short time, but very large change in the membrane potential from, it is 0 here to plus 30, and very rapid. This is called as action potential. It involves only a small portion of the total excitable cell membrane. It will not decrease in strength as it travels from from their site of initiation throughout the remainder of the cell. Suppose this is the whole cell. Suppose this is the size of the cell. I'm drawing it in a rectangular fashion, but it can be a circular also. Suppose there is an action potential here. So it will not become less, and less like this. No, it will be, this is not possible in action potential. This is not possible in action potential. How it will be? I will show you here how it will look like. Suppose this is the whole rectangular is a cell. So if the action potential is reduced here, it will be same in the other side, traveling to till the end. Same height, almost same height it will be there. It is not like decreasing, huh? Not like this. So do not decrease in strength when it travels from one area to other area. This is a property of action potential. So this is a graph of action potential, which shows you this is 1 millisecond time. See, this is only 1 millisecond time when the membrane is changing its potential, and it is changing from minus 90 millivolt till plus 30 millivolt. Very large amount in change. Now you will see that there is a green line. What is this green line means? Green line means it is a threshold potential. Threshold potential. What does it mean? It means that when the potential of the cell membrane is reached to a minimum level, required level, suppose minus 70 to minus 50, if it goes, it will start to have action potential. It will start to have action potential. If the membrane potential goes from minus 70 to minus 60, it will not have. It will not convert it into action potential. So threshold means the minimum required change. Minimum required change in membrane potential. This is called as threshold potential. The minimum required change is here in this example, it is from minus 70 to minus 50. That means minus 20 millivolt change is there. In this example, it is minus 20 millivolt change should be there to bring the action potential. This is called as threshold potential, right? Any question?
Yes, Doctor. How do I know the minimum required change in action
Yes.
potential?
How do you know the minimum? You cannot know the minimum. For each cell, it will be different.
So then how do I know the threshold of potential?
No, it will be shown to you in the graph.
Okay.
In the graph only the line should be shown. That is called as the threshold for it. So anything below, you will say there will be no action potential. If it goes and reaches here, then there will be action potential like this. Okay.
Okay.
So another name for action potential is also known as spike potential. Spike. Spike means like this, masmar. Because it's very sharp, pointed. So if it is recorded, it will be like this. That's why it is known as spike potential. Another name for action potential is its spike potential. Another terminology is used in excitable cell, that when action potential is produced, they say the cell is firing. The cell is firing. Firing. When it is firing means it's producing the action potential. It is producing the action potential. So action potential or spike potential or firing of the nerve is this one and the same thing, which is referred to as action potential. So action potential, the cause of action potential is mainly the voltage gated sodium and potassium channel. Voltage gated sodium channel and voltage gated potassium channel, their activity, their opening and their closure. Why it is called as voltage gated? Because they will open and close based on the voltage of the membrane. Based on the voltage of the membrane. If the membrane reaches to minus 55, then they will open. Or they reaches to minus, like here, if they reaches to, the cell membrane reaches to plus 10, maybe 10, 20 millivolt, then they will open like this. So they depend upon, they open and close based on the voltage. That's why they are called as voltage gated channels. So action potential is produced in this fashion in the form of a positive cycle. Start with opening of some of the voltage gated channel, which will cause opening of, this is the threshold minimum required. then it will lead to more opening of sodium channel. This will lead to positive, the negativity inside will become positive. We call it as depolarization. Once depolarization occurs, it will again cause opening of few more sodium channels. So like a positive feedback mechanism, it will create more change in the membrane potential. So this graph is very important because it is summarizing the whole thing. This graph is very important because it is whole lecture. The whole full lecture is can be summarized on this graph. So what is this line? Straight line. What do you call this line?
The depolarizing
No, this is straight line.
threshold.
Here it is written.
Resting
Resting.
potential.
Yeah. So whenever it's straight line is there, it is resting membrane potential. Okay. When it goes above and reaches to this dotted line, what is this dotted line means? Threshold,
Threshold.
minimum requirement for the action potential to produce. So when this goes up till here, it is the threshold. After the threshold, sorry. After the threshold, it suddenly moves up because many more sodium ions are open, channels are open now. Sorry, many
So this
sodium
graph,
channels are.
this graph might come in the exam.
Of course,
Okay.
I told you graphs and figures are important because they can come in the exam.
Okay.
So from here, it is suddenly changing its potential very fast and very rapid and going across, crossing the zero line and moving up. What is the cause? It is the sodium ions moving into the cell. Sodium ions are moving like this here. If it is a cell, so more sodium ions are moving into the cell, which we call it as sodium influx. Sodium moving into the cell, we call it as sodium influx. Because of the sodium influx, the cell is becoming more positive, and that's why it is going towards zero, losing its negativity, and it is towards zero. So sodium influx or sodium inward movement, influx or inward movement. If, as we say, these sodium channels are voltage gated. So if the cell membrane reaches to the a point where it is plus 20 or plus 30, suddenly the sodium channels will close. These sodium channels will close and potassium channels will open. Now at this point, if it is plus 30, potassium channels will open. Once the potassium channels will open, here is the potassium channels, and this is inside and this is outside, it will cause potassium to move out. Potassium to move out. Outward movement of potassium. It is also known as efflux. Outward movement or efflux or exit of potassium. The potassium is out now here. Out, going out. And that's why the membrane which was plus now, it is going towards the negative. Reaches to the threshold, but it will go more negative. Not only minus 90. Here it is minus 90. It is becoming minus 100 maybe. Why? Because these potassium ions which open, they close very slowly, slowly. They are moving. And that's why more potassium will be lost. More potassium will be lost. So sodium potassium pump, after the finishing of this, It will restore the same concentration of sodium and potassium into and outside the cell. So at resting membrane potential, again, sodium potassium pump will be active. And action potential is not local potential. Once it is produced at one point, it will be traveling to the other areas also, from one area to other area, from one part of the nervous system to other part of the nervous. This is called as traveling, not local. Huh? Okay, one more thing is that related to action potential is that it follows or obeys a law which is known as all or none. All or none law. What is all and none law?
يعني in or not in يعني.
Yes. All or none means either there will be action potential or there will be no action potential. There cannot be a small action potential or a big action potential or very big action potential. If there is action potential, it will be like this only. Same height, same width. You cannot say
Doctor,
that I stimulated little bit, so there will be little action potential. I stimulated more, so there will be more action. No, it's not possible. Not possible. Either there will be action potential or no action potential. That's it. This is called as all or none law.
according to the trigger event.
Yes, even the trigger event, it has to reach to the threshold. If it reaches to the threshold, there will be action potential. If it does not reach to the threshold, there will be no action potential. You cannot say that it is not reaching to the threshold, so it will be smaller potential. No, there will be no smaller potential. Okay.
Doctor, when I say decrease in the membrane potential, it means like being more positive.
Yes. You are losing the negativity. Yeah.
Okay.
Now coming to the two time periods, which is called as refractory period, absolute refractory period, and relative refractory period. This is related to the action potential. So again, coming back to the graph. So this is an action potential. If you stimulate the cell here, then you will find the action potential. Okay. If you stimulate it here at this point, so from here, you will have starting of the action potential. If you stimulate it, second stimulus is near second after first one. This is number one. This is number two. If you stimulate twice, two times, you will not find any second response. You will not find any second response. Or three times also, there will be no second response. Why? Because the cell is in refractory period. Cell is in refractory period. So there will be no second action potential if you stimulate it close. One stimulus is here, second is here. It's still you will get one response only. Okay, this is called as absolute refractory period. So from here till here, till this point, if you stimulate any time, you will not get second response. You will not get second action potential. This is called as absolute refractory period. What is mean by relative refractory period? Relative refractory period means, again, I will show you here the definition now. It is the period during which the second action potential can be
Doctor,
produced.
the screen freezed again.
Again, why?
It's still in the action potential picture.
Okay, okay. Thank you for telling me. Same graph you are seeing, huh?
We're seeing the other period now.
Can you see now the definition of absolute and relative?
Yes.
Yeah. So absolute refractory period is the period or the time in which no second action potential can be generated. If you stimulate it here or here or here, there will be only one action potential. Whereas relative refractory period is the period in which if you stimulate a second action potential can be produced by very strong stimulus. If suppose you, instead of stimulating here, you are stimulating here at this point with large magnitude, maybe 100 millivolt current. If you are stimulating here, then you will find second action potential. Then you can find a second action potential. This is called as relative refractory period. So if you see this graph, it will show you that from the point of starting of the first response till the end or till the middle of the repolarization phase up till here is the absolute refractory period. Maybe if you have some stronger stimulus after this period, you can have relative refractory period. That means second action potential can be produced if you stimulate in the later 1/3 of the, I will show you here. So from here till here, or maybe up to here, sorry, up till this point, there cannot be second action potential. There cannot be second action potential. That's why it is called as absolute refractory period. But if you stimulate the second time in this period from here to here with a strong stimulus, not the normal one, strong stimuli, then there can be second action potential can be produced. So this period is called as relative refractory period.
Yeah, Doctor, absolute, no potential, relative action
No
potential.
second action potential. Okay. By any means, by 100 millivolt current, 200 millivolt current, 1000 millivolt current, there cannot be second action potential. In relative, yes, there can be second action potential if the stimulus is stronger. Getting my point?
Yes, only produce action potential, strong stimulus.
Yes, good.
Okay.
Why it happens? Why it happens? Because the sodium channels which open to cause sodium ions to move in, they cannot close and reopen again in this absolute refractory period. They cannot reopen. Once they close, they will take some time to reopen again. But in relative refractory period, maybe out of 100 channels which are closed, maybe 10 will start to open again. So there can be second stimulus can be produced here. That's why there is a relative refractory period. Done. So this is a summary of the whole thing which I talked about action potential. Action potential is a regenerating means repeating. Regenerating means repeating. Depolarization of the membrane potential that will propagate. Propagate means travel from here to here to here to here till the end. Till the end. And you can see the same height. Huh? It is not decreasing. It is not decreasing. So this is called as action potential. All or none. All or none means when threshold is reached, there will be action potential. If threshold is not reached, there cannot be small or big action potential. It is either present or absent. There cannot be small or big. Okay. All the amplitudes are constant. all of them amplitudes are constant, same height. They cannot be added together. You cannot say that this is one action potential, this is another action potential. If you join them together, there will be big action potential. No, this is not possible. So they cannot be added together. They are initiated by depolarization. It involves the changes in the permeability, and it is based on the voltage gated ion channel, sodium voltage gated sodium channel and so voltage gated potassium channel. So if this is the graph of action potential, so now tell me which channel will be active in this phase, depolarization. Which channel will be
Sodium.
open in this phase? Tell me now.
Potassium.
In
Sodium.
depolarization, which channel will be open?
Sodium. Sodium.
In sodium. And in repolarization?
Potassium.
Potassium. Potassium. Here, sodium is moving where? In or out?
Out.
Out.
In depolarization, sodium will be moving
N.
inward, influx. I told you this is influx. And here potassium is moving
In.
Out.
مدري out. Out.
efflux or outflux or outward movement.
Okay. So, Doctor, can you repeat this thing again, please? يعني depolarization for sodium, moving inward.
Yes, like
Okay.
this. Suppose this is a cell. So sodium is
Okay.
moving in.
Okay.
Many sodiums are moving in now. Okay.
Okay.
Now in this phase, for repolarization, sodium is already there, but this will not go out. Instead of that, potassium will go out. Potassium will go out. Okay. To balance.
Okay.
So influx of sodium is here and efflux of potassium is here. And what is happening here? Let me show you. The one where hyperpolarization is occurring. Okay, here sodium is in. Here potassium is out. What about this phase?
Hyper.
Hyper.
Yes, this is hyperpolarization. What is happening here? Is it sodium? Is it potassium? Or is it something else?
Something else.
Huh? Something else. Which one? We did not discuss any other ions, only sodium and potassium.
More potassium leaving the cell.
Huh?
More potassium leaving the cell.
Yes, continuous potassium moving out of the cell, potassium out. Suppose I will give you example. See here. Here, let's say 100 sodium moves in. Okay. Here, 100 potassium moves out. And here, maybe more 10 potassium moves out. Excessive potassium moves out. Why? Why it is so? Because if you might have seen, let me clear all these things. Have you seen a villa where a big gate is there? Let's say this is the big gate of a villa. This is a big gate of a villa. This is the gate, huh? Once it open, the cars come out. But once it close, it will take slowly to again close. So by that time, more people will go out. So 100 sodium moves in, 100 potassium goes out, but here more potassium will go out because it takes long time for closure of potassium channels.
That's for hypolarization.
Hyperpolarization. Hyperpolarization occurs because of slow closing of potassium channel.
Okay.
So instead of 100 sodium, 100 positive ions which are moved in, 100 potassium should go out. No, now instead of 100, 110 potassium goes out. That's why it is going below.
Okay. Oh, repolarization, sodium is moving up.
Yeah,
Doctor?
again, the same thing will go on.
Okay. Thank you.
So this is the slide showing you the difference between action potential and graded potential. What is the difference? Difference potential is the stimulus does not reach to the threshold like this and come back. Here, action potential, the stimulus has to reach this line. Then only there will be action potential. Okay. Always, whenever there is action potential, that means the stimulus is reaching to the minimum required level, that is threshold. The stimulus causes local change in the membrane potential. Let's say from minus 70, it is going to the minus 60 only, small. Here, stimulus depolarization at the threshold level will occur to minus 55 or something like that. It will be dies down over a short distance. It is starting here, and then there will be no. So after some time, you will not find any membrane potential. Here, if it is found, it will travel to the whole length, propagated. It's propagated throughout the length of the neuron or a muscle. Here, graded potential can be added. 1 millivolt change plus 1 millivolt change will give you big 2 millivolt change like this. So it is can be added. Here, you cannot add this plus this will give you this. No, not possible. Not possible. Does it obey all or none law? No, it will not obey. A small current, a small change. Big current, big change. Here, it obeys all or none, either threshold and action potential, or no threshold or no action potential, like this. This is called as all or none law. All or none law is for action potential. Now tell me, what is the difficulty?
Doctor, can you please repeat the refractory period?
Yes, refractory period is the period where, suppose I stimulated, I have to show it on the diagram. Let me show you on one of the slides. Okay. So, yes. I am drawing it slightly bigger because it is not like a hum. It is like a sharp pointed edge. Okay. So if I stimulated the neuron here at this point, let's say 1 second time.
Doctor,
Okay.
the screen is freezing. Sorry.
Yes, okay.
Doctor, if you after you finish explaining refractory period, is there any information or extra things that you want us to are you going to tell us, or خلاص, this is the end of the
No,
tutorial?
end of it. No extra. If you want
ما
to
في
leave, you can
tips?
leave.
Is there any tips منا يا دكتور? Any information?
No extra information. Same thing. I explain everything now.
Hint يا دكتور, no hints.
No hints.
No
What
no
hints you need?
questions يا دكتور. Example question يا دكتور.
Example questions. I already put it on the Moodle. If you remember, on Moodle, there is a one question, one at the end.
Question exam.
Yeah, some different type of questions are there on the based on the exam. You can do.
Okay, thank you, Doctor. Thank you so much.
Where is the whiteboard? Yeah. Can you see the whiteboard?
Yes.
Okay. Oh, where's the pen? Color. All this. So this is a diagram of action potential. Okay.
Okay.
When we start stimulating the neuron here, the action potential is produced. If you stimulate it here again, then you cannot have a second graph. You cannot have a second graph. Why? Because from here till the last 1/3, till this period, if you stimulate so many times, even with a big stimulus, very high voltage current, still you will not have second graph. This is called as refractory period, absolute. Why? Because at this time, the channels which are responsible for moving the sodium in, they are already in the closed state. They cannot allow any sodium ions to move in. Okay. That's why the second graph is not possible. We call it as absolute refractory period. Absolute. Not possible by any means to open these channels again. In the last 1/3, and this last 1/3, if you apply a very strong current, very strong, maybe suppose here you are applying 10 millivolt current, suppose. If you apply it 100 millivolt current here, then you may have second graph. You may have second graph because what is happening? Let's say these are the channels which are closed now. Closed now. So like this, they are closed. But by reaching here, these channels might have opened now. Might have opened some of the channels, which will allow sodium to move in. And that's why it is possible for the second graph to occur. This is called as relative refractory period. You got my point?
Yes, doctor.
In
Thank you.
absolute refractory period, you cannot stimulate the nerve again to give second response. But in relative refractory period, if you apply a bigger current, very strong stimulus, you may get a chance to have a second stimulus. This is the only explanation of absolute and relative refractory period. The duration in which you cannot have second stimulus, this is called as absolute, and the duration in which you have chance of having second stimulus or a second response, this is called as relative refractory period. And I gave you the reason. The reason is in relative refractory, in absolute refractory period, sodium channels which are closed cannot be reopened. Cannot be reopened by any means. If suppose the shop is closed now, it will open in the morning only. Not you cannot open the shop in the midnight or just at Fajr prayer. So the shop is closed, means closed. You cannot buy anything. In relative refractory period, no, some shops can be open around midnight or around Fajr prayer. So you can buy something. So this is the explanation, any example I'm giving you, that if there is closure of all the channels, you cannot reopen it and you cannot have second response. But in the later some part, you can open some of the channels, so you will get a second response. This is called as relative refractory period.
Thank you, doctor.
Okay, well. Any other question?
Thank you, doctor.
For practice, you can see the practice question. It will be good for you to practice it on the Moodle. What type of question can come in the exam for graded potential and action potential?